Aloha, everyone, and thank you for joining us on your lunch hour to learn more about a parasitic disease. Always a good time over your brown bag. I am Franny Kinslow Brewer, and I am the Communications Director for the Big Island Invasive Species Committee. This is our last Hawaii Invasive Species Awareness Month webinar for February, but our partners across the state have a few events still planned for you. Today at 2 p.m., our friends over on Molokai will be talking upside down jellyfish control, which sounds fascinating. And tomorrow at 4, our ISC partners over on Hawaii will be tackling the question, is ohia worth saving? So that sounds like that's going to be a pretty robust discussion. And of course, on Monday, the final day of Hawaii Invasive Species Awareness Month, we have our closing ceremony at lunchtime. You can find information about how to join into all of those events on the link on the HISC website, and that's going into the chat right now. I would like to point out that we have some folks supporting us here today. My BISC team, Jade is on Zoom. Say hi, Jade.
Hi.
And she's the one going to be putting these links in the chat. And then over on Facebook, where we are live, we have Molly. Molly, say hello.
Hello, everybody.
So they are going to be watching, monitoring the Q&A and the chat. If you have any questions, you can go ahead and put that in there. And they're adding links right now to the places I'm talking about. So if you want to find some more information about those webinars that are being offered for the rest of the month, or the next few days, you can go to that first link. And if you look at that link and you see a whole bunch of talks from a couple weeks ago and you're thinking, oh man, I really wanted to see that, I want to miss it, don't worry about it. We have been recording all of these, and you can find any of those on the HISC YouTube. Link is going in the chat and up on Facebook now to get to those. And you can go there and watch any of the recordings. We will also be posting the ones that BISC has hosted on our BISC YouTube. So I'm going to plug for that because we have a lot of other great information on there about two-line spittlebug, how to control little fire ants, all kinds of good stuff on there. So please go and check it out. Jade is going to be launching a poll here, just very quick, couple simple questions. If folks would not mind answering those few questions, that really helps us kind of get a sense of how these webinars are, the audience that we're reaching. And if you're over on Facebook, you don't have access to the poll, just type into the chat what island you're watching from today. And Molly will take care of recording that. So really appreciate it. Just giving it a second to let everybody do that. And I want to also start by introducing our guest today. We're really excited to have our friends from NOAA here, the Monk Seal Recovery Team. They are going to be sharing with us what they've learned about toxoplasmosis in monk seals. That is, as I referenced when we first started, a parasite well known for causing sickness in a number of mammalian species, and I think some birds as well, but we're really concerned about how that impacts our endangered monk seals. So first up, I want to introduce Angela Amlin, who is the Hawaiian Monk Seal Recovery Coordinator for NOAA, for the National Marine Fisheries Service. She has spent the last 14 years focused on conservation and recovery of threatened and endangered species, working in multiple sectors and with diverse taxa, including seabirds, water birds, bats, and now pinnipeds. So she currently is sharing her home with 2 very spoiled cats, but they are indoor cats. And I imagine that some of that is driven by the things that Angela knows from her day job. So Angela, thank you for being here with us today.
Thank you, Franny. Wait,
and I know,
just a second. Oh, sorry.
Oh yeah, I was gonna say, I know you have like a whole series of slides, so you are free to share your screen whenever you're ready, and we'll be looking forward to hearing that.
Okay, 1 second here.
Angela, you are still muted, and your screen is black, so I'm not sure a little bit of a technical. Do you want to unmute yourself?
Says that my it's going to mute when I share, but when I shared earlier, that didn't happen. That is strange. Let me try this one more time. I apologize.
Okay, that is very odd because it did mute you again. And we just tested this. So I don't know.
We did. Great. I'll tell you what I'll do. If you want to go ahead and introduce Stacie, let me see if I can switch over to PowerPoint from Google Slides. Maybe that'll work better.
You can totally.
Sorry, folks. There's always technical difficulties.
And then we get a chance to see Stacie's smiling face. Stacie is a research ecologist with the Hawaiian Monk Seal Research Program. She has a PhD in wildlife ecology with a focus on wildlife disease research. And Stacie's worked as a research ecologist for NOAA's Hawaiian Monk Seal Research Program since 2014. And I've had the pleasure of working in a working group with Stacie for the last year. So this next part, that she is passionate about protecting Hawaii's native wildlife from disease and other threats. I have seen that in action. So glad that you could be here with us, Stacie. Really appreciate it. Let's say hi to the folks back home.
Thanks, Franny, and thanks for everyone for joining us today to talk about this really important topic.
Yes. Angela, do you want Stacie to go ahead and give her presentation, or would you want to try to, I think you're ready to try to show the screen.
Yeah, our presentations go in order, but Angela, if your next attempt doesn't work, I could always screen share, and you can tell me, you know, when to push buttons to flip through your slides.
Okay, it's opening right now. I'm sorry, it's slow. I'm on Zoom. You'd think we would have this all figured out after 2 years of this. So thank you so much for your patience.
And I promise, folks, we tested this at about 12:20 and it worked fine. So
just fine.
Sometimes technology just sneaks up and bites you.
The patience.
All right, let's try this. Is
Yay.
that?
Yes, perfect. Wonderful.
Okay, fantastic. All right. So sorry about the false start there. So I did want to say too, for those of you who joined on, I think it was Tuesday night, a few of the slides that I'm going to be sharing might be repeats for you because it's a strong overlap in the topic. So I apologize for that. But there is a lot of great new stuff in here, especially coming from Stacie and the research side of things. So please bear with me as well with some of this repeat. So again, really quickly, we'll just do a background on Hawaiian monk seals for those who might not be aware or who might not be familiar with them. Hawaiian monk seals are endemic to the entire Hawaiian archipelago. There are about 1,400 of them left. The vast majority, about 1,100 or so, are in the northwestern Hawaiian Islands or Papahanaumokuakea Marine National Monument. There are about 300 to 350 in the populated main Hawaiian Islands, with Ni'ihau having the largest population in the mains and the Big Island having the smallest. They are protected federally under the Endangered Species Act and the Marine Mammal Protection Act, listed as endangered and depleted respectively. And they're also protected under state law under Hawaii Revised Statute 195D. These guys are fairly long-lived. They live to be about 25 to 30 years of age. They don't start reproducing until they're about 5 to 6 years old in the main islands and a couple years older than that, between 7 and 10 in the northwesterns. They have only one pup per year, and they don't necessarily pup every year. Twins are exceedingly rare, and the number of pups that they have does decline with age. So when did toxoplasmosis start to become, and I'm sorry, with the download, it looks like some of my graphics got a little weird here, but so apologies for that as well. So when did toxoplasmosis really come onto our radar as a threat to Hawaiian monk seals? It was really in 2015 when this became an emergent concern for us. That's when we had the first live stranded cases of monk seals with toxoplasmosis. There were 2 of them. Both of them we were able to bring into our facility, and we had hoped to start treatment, but unfortunately both of them died within 48 hours of admitting them. This was, however, unique because most Hawaiian monk seals with toxoplasmosis we'd find dead. This seems to be something that hits them, and when it kicks in, it progresses very quickly. So this was unique, and it taught us about a behavior called logging that is pretty much our only key to early detection. Logging is kind of what it sounds like when a seal floats listlessly in the water like a log, and it just sort of lets the tide move it. It's not really moving of its own accord very much. I do want to caveat that and say just because a seal is logging doesn't necessarily mean it has toxoplasmosis. They might do this when they have other minor injuries just to save energy. We have some pregnant females who do it, and we do have a couple seals who like to do it just because they like to throw us through loops. But having at least this one behavior that we can key in on is helpful for us to at least try to intervene as soon as we can. These cases also illustrated just this massive inflammatory response to infection that monk seals have. This can infect any warm-blooded animal, but animals that have co-evolved in ecosystems where cats are native seem to maybe have a more robust immune response than monk seals do. And so around this time, we elevated our level of concern about this threat. And we actually went back and we looked at some previous necropsies where the results had been inconclusive, and we found some additional cases. And I'll show a little bit more of a timeline in just a second, but I will say that since these two patients in 2015, we have had 2 more monk seals that were brought in alive. Both we were keyed into by a report by a community member of a seal logging. And then we brought them in for treatment. One was a female, one a male. One survived for 10 weeks and one survived for 6 weeks. So no success stories yet, but we're still learning a lot and attempting to improve our treatment protocols. So looking at this seal timeline here, since 2001, there have been 15 documented deaths as a direct result of toxoplasmosis. This number does sound small, I know, but given that there's only about 300, 350 seals in the main Hawaiian Islands, it is actually a fairly large percentage. And we don't recover every carcass. I think we recover close to, but maybe just shy of half. Many others are washed out to sea, eaten by sharks, and even some of the ones we do recover are not in good enough condition to test. So we know that 15 is an underestimate. Of those 15 deaths, 10 were female. Those are the seals you see up on screen in red. So it seems to be disproportionately affecting females, and I'm sure you can guess why that is important, and I'll talk a little bit more about that in a minute. The blue seals up there are males, and there are 2 yellow seals. Those are not included in the 15. Those are suspected toxoplasmosis cases. We know they died of a protozoal infection. Toxo is a protozoal parasite, but we could not definitively conclude if it was toxoplasmosis, and so therefore we do not include suspected cases in the count. We only include confirmed cases. Another way to look at this is looking at it spatially. You can see that by far the most deaths occur on Oahu, followed by Kauai at a bit of a distance. This makes sense. Oahu has the biggest human population in the mains, and so therefore it's going to have the largest outdoor cat population, or at least a large outdoor cat population. Kauai has the 3rd largest human population, but it has far more seals than Maui does, which has the 2nd largest human population. And so therefore we also have really robust sightings networks on Oahu and Kauai. This is networks of people who call into our marine wildlife hotline any sighting of seals, regardless of their health status. But very importantly, seals that appear to be sick or injured. And I would encourage folks listening today to do that as well. I've put our hotline number 888-256-9840 up on the screen. And that way we can be alerted ASAP when a seal is sick or injured, and then we can respond to it as quickly as possible. I'm sure you also notice up there, there's a lowly little lay sand seal up there, which is interesting and kind of scary. We don't know exactly how that happened. Did it contract the disease in the mains and then return to the Northwesterns? They do swim back and forth. Bilge water in a ship, long distance ocean transport, a prey item, we just don't know. Between 2015 and now, this issue has garnered an increasing amount of attention, which is good in many ways because it brings awareness to the issue. It gets information out there for public consumption, and it can be a mechanism for reaching populations who may not have been aware, or segments of the population who may not be aware of this issue previously. But it does have drawbacks. It can be divisive, and it can amplify existing conflicts. You can see the crazy war over zombie cats article up there in the middle. So how is toxoplasmosis affecting our endangered monk seals? Toxo is the primary disease threat far and away in the main Hawaiian Islands to monk seals. And it is the third leading cause of death for seals in the mains overall, following anthropogenic trauma, which is intentional killings, drowning in nets, and then you have toxoplasmosis. I will say that this chart is, or this bar graph is from Harding et al., which was published in 2020. So the data are a few years old. There's unfortunately been a few more deaths in each category, but that ratio's staying about the same. So it's still looking at the third leading cause of death. The impacts of toxo are especially heavy for monk seals because, as I mentioned before, and as you can see in this graph, toxo hits adult females especially hard. And because it can be passed from mom to pup transplacentally, it impacts fetuses as well. And so when an adult female dies, we don't just lose that one seal, we lose her lifetime of breeding potential. One of the females that you saw on that timeline was a first-time mom. There were two, I think, female fetuses. So we don't know how many seals they would have gone on to have, and how many of those would have been female, and how many pups they would have gone on to have. So when we're looking at an endangered population, that's quite significant. And that Harding et al. Paper found that toxo, as well as those other threats that I mentioned, are definitely limiting potential growth of the main Hawaiian Islands population. So to recap a little bit some of the key points, these seals are seldom found before they've actually succumbed to the disease, although we are working really hard at disease surveillance and at early detection and working with the community to report sightings as frequently as possible. Our treatment options are very limited and thus far have not been successful. So what we can conclude from this is that treating monk seals is not the answer to the solution. That's not the best approach here. While we will continue to invest resources into doing so, what we really need to do is mitigate the impact by limiting pathogen exposure. And with that, I'll turn it over to Stacie to talk a little bit more about the basic biology of toxin.
All right, thank you, Angela. I'm just going to share my screen and we will carry on with the presentation. Let's see. Okay, so I guess anyone let me know if there are any issues with the visuals or the audio, but it looks okay on my side. So as Angela mentioned, we really need to focus in on controlling the disease agent and the environmental contamination with this pathogen in order to control the threat or mitigate the threat for Hawaiian monk seals. So one of the first things we want to do is really think about the basic biology of that organism to sort of understand the enemy and understand what we're dealing with. So one of the reasons that we're in this invasive species awareness month talk is because felids, or here in Hawaii, that's going to basically just mean domestic cats, are what we call the definitive host of the Toxoplasma gondii, the parasite that causes the disease toxoplasmosis. And so what we mean when we say definitive host is that cats provide the only reproductive environment where this parasite can sexually reproduce. And so that actually happens inside the cat's gut. You have the male gametes from the Toxoplasma organism and the female gametes, and they come together to form an oocyst, which is kind of a funky word, but it basically is like an egg, except you can think of it as kind of having almost being armored on the outside so that it's extremely hardy in the environment. So these oocysts can actually persist for years in soil or in water. So they're out there in the environment and can remain infected for a very long time. And so after this reproduction happens in the cat's gut, then those oocysts can be passed into the environment through the cat's feces. So where does the rest of the life cycle come in? So we've got cats with feces delivering oocysts into the environment. And then what happens next is that other animals can pick up those oocysts, say if they're eating grass that's been contaminated with cat feces, or insects or whatever. And so animals like birds and mice are considered intermediate hosts because they will consume these oocysts, and then the oocysts can form tissue cysts in these animals' bodies. So it's a different life stage of the Toxoplasma parasite. And then we complete the life cycle when cats eat these animals. The cat ingests those cysts, and then the reproduction can happen in the cat's gut all over again. So there's another sidebar of this life cycle, though, where other animals can also become infected. Toxoplasmosis can pretty much infect any warm-blooded animals. So that means birds and mammals are at risk. And so there are a whole other host of what we call intermediate hosts, which are basically any other animals that aren't part of this life cycle getting consumed by cats, but they can also get toxoplasmosis or toxoplasma infections. And like Angela was saying before, depending on sort of the nature of the animal, maybe whether they co-evolved in an environment where they're used to cats and cat parasites, different animals are affected to different degrees. I'm sure you've probably heard things about some of the impacts to human health. Toxoplasmosis has been associated with certain types of blindness and pregnancy complications with women. So if you've ever heard, pregnant women shouldn't change the litter box, this is the disease that that folks are talking about with that. So it can be a concern for human health. It's been found in some of the Hawaiian spinner dolphins. It's been found in nene, in alala. So basically all of those warm-blooded creatures can be susceptible, and in some of them it can be quite harmful. And so just to kind of reiterate some of the key differences, and especially what we mean by intermediate versus incidental hosts. So these incidental hosts are ones that can get infected with toxoplasma, but they're not getting consumed by cats. So they're not part of that life cycle of the parasite. They're kind of a dead-end host from the parasite's perspective, whereas intermediate hosts are the ones that actually get consumed by the cat and allow the parasite to complete its life cycle. And then another key difference, so even though we've just said basically all these warm-blooded animals can become infected, because the parasite is not reproducing in any of these intermediate or incidental hosts, they're not producing any of those oocysts, so there are no eggs going out in the feces. There's no environmental contamination happening from any of these hosts. And it's just that definitive host, the cat, where oocysts are produced and then can be shed in the feces to lead to environmental contamination. So the next kind of important step is, once we understand sort of this, this life cycle, the oocysts being shed in cat feces and the contamination that can happen in the environment, then what we want to do is find links from, explaining how that contamination gets from the terrestrial system where cats live to the marine system where monk seals live. And so what we do here is we take kind of an epidemiological approach using what we call the epidemiological triangle, where we try to understand the pathogen, the environment that the pathogen is existing in, and then how the host might encounter the pathogen in that environment. And so one of the first things we looked to do was to investigate the correlation between heavy runoff events and known cases of toxoplasmosis, to see if there might be signals that major runoff from the terrestrial system was bringing oocysts into the monk seals habitat and infecting them. So what we did was we set up a case control study. So that's basically where we compare cases or animals that stranded with toxoplasmosis to animals that are what we call controls, which are animals that stranded or died from reasons, other than toxoplasmosis. And so in this map, you can see we've got both cases and controls on Kauai, Oahu, and Molokai. So these are basically all of the toxoplasmosis deaths that we had from 2005 to 2020. We ended up with 8 on Oahu, 3 on Kauai, and 1 on Molokai. And then we sort of matched control seals, or seals that died of other causes, for that same time period in the same islands. So then what we wanted to do was compare the runoff exposure that those animals had. So we looked at stream gauges in the regions of each of those seal deaths. We looked at stream gauges in the waterways that were feeding the areas where each of those animals was found. And we looked at stream gauge readings for, up to a couple of months before that stranding event. And then what we did was we compared cases and controls to see if they had kind of similar patterns of exposure to runoff, or if potentially toxoplasmosis cases were exposed to larger or more major runoff events than the controls. So what we did was we looked at standard deviations from the mean, or the average. So this 0 represents basically the average level of runoff from any given stream gauge that we looked at. And the gray box is 2 standard deviations above and below that average. So basically that gray box bounds what we would consider all within the normal range of runoff. We're not going to really, it's not piquing our interest if the runoff is within that gray box. So what we're really interested in are these instances where we have these black dots above the gray box that show a major runoff event, or one of those, beyond, twice the standard deviation from the average. And we do see a couple of those in our controls, but we see a lot more of those major runoff events in the case seals. We see, so for each seal, we see more seals with those dots, and we see more dots for those seals, and the dots reach higher, indicating they're even, like a greater magnitude above the normal for runoff. So this starts giving us an indication that probably runoff is actually a factor contributing to the risk for these seals that got toxoplasmosis and stranded and died from that disease. And so then we looked at it statistically using what's called an odds ratio calculation, where we look at the odds that a toxoplasmosis case seal was exposed to a major runoff event versus one of these control seals being exposed to a major runoff event. And what we found was the most significant odds ratio is this peak at about 3 weeks, where the toxo cases were about 35 times, had 35 times the odds of being exposed to a major runoff event versus the controlled seals. And then we also did some additional randomization tests, and all of these yellow stars show weeks in which those were showed significant differences between the cases and controls as well. So what we could see from all this is, when we ask the question, does terrestrial runoff increase the risk of toxoplasmosis exposure to monk seals? The answer is, it's really looking like a yes. We did see greater association with major runoff events for those cases versus the controls. And really this finding is not too surprising. It's totally in line with what's been found in other ecosystems, for example, on the West Coast with sea otters that get exposed to toxoplasmosis in that environment. And so this also supports the role for surface runoff in oocyst exposure, and really helps demonstrate that local link between the pathogen source and the exposure of our host of interest, the monk seals. So we can kind of see, the local process happening with our local contamination runoff into the local environment. But, that also gives us some hope that some of the solutions and mitigations for this risk can also be quite local. And then it also, seeing that peak at the 3 weeks prior to stranding point, that gives us a potential clue to the epidemiological timeframe, or the timeframe that it takes from exposure to the pathogen to the outcome for the seal. And of course, this is something that we'll do more examination with veterinary medicine, but it's cool that this data can at least kind of point us in a direction to look at a little bit more there. And so then the next thing we wanted to do was, okay, now that we understand there is this pretty clear relationship between the runoff, potentially bringing this terrestrial pathogen into the monk seal's habitat, we wanted to do some work really mapping the risk of where that runoff might be happening the most, and try to figure out how we can best mitigate that risk. And so again, turning to our epidemiological triangle. So in order to develop this risk model, what we're going to do is we're going to start with thinking about oocyst loading, or basically how much of the pathogen is where on the environment. And then in thinking about the environment, we're going to be using hydrological models to think about the transport. So like, how is the hydrology of the island bringing those oocysts from the terrestrial into the marine environment? And then of course, thinking about where seals are, to think about the exposure of the host. And so with that first step, thinking about the oocyst loading. So in the literature, there's a lot of information about, things that you might never think someone studies, but like the average defecation rate of cats. And so, how much cats are defecating, and then the rate at which cats are infected with toxoplasmosis. If they're infected, how many oocysts they shed. So there's that information in the literature that we can calculate about the oocyst from any particular cat and its probability of being infected. But then what we really need to know is how many cats are there and how are they distributed across the landscape. And for this, one of the first things that we wanted to do, or felt like we needed to do, was really sort of categorize the cats to think about like, well, there are different types of cats. We have different amounts of information on them and different sources of information on them that we can use to put into this model. And so we divided cats into 4 categories based on their relationship with human beings. And that, of course, impacts the cat's ecology and the likely habitats that they're using. And the habitat is important because that gives us the ability to map where cats might be based on some of the ecological habitats that we have on Oahu. And so first we had companion cats. And so an important thing to remember here is when I talk about companion cats, it's just the portion of companion or pet cats that are allowed outside. So with all of these toxoplasmosis considerations, we're really just concerned with cats that are able to live and defecate outside, right? Because if a cat is inside using a litter box, then that's taken out of the risk landscape. So here we're talking about outdoor companion cats. And then colony cats, so cats that are pretty associated with people. They're getting fed by people, but they're living outside, kind of free roaming in the environment, but still have a very tight link to the people that feed them. And then next is stray cats. So these are cats that are, still living in town or maybe in human developed environments, but not directly getting fed by people, but might scavenge off of, garbage or human related food resources. And then finally, we have kind of our most wild cats, the feral cats. So these are the cats that are free roaming in the environment, but away from human settlements. So relying more on natural prey to get their dietary needs met. And so once we had these 4 different types of cats defined, we wanted to think about where on the map of Oahu these different cats would go. So we took just some basic landscape characteristics and landscape zoning for the island of Oahu. And so we kind of split up the island based on urban versus agricultural versus conservation areas, also took into account developed and non-developed areas with and without roads, and whether an area was of extreme slope or open water. And then we kind of created a rubric of rules of where these different types of cats would go. So you can see companion cats would be around urban areas and agricultural areas, but we don't expect companion cats to be out in the undeveloped conservation land. And of course, we don't expect any of the cats to be on, a completely almost vertical slope or in open water. So once we had sort of our landscape of where cats could go, and we relied on a number of different, surveys and publications to figure out how many cats of different types, and we varied the numbers of cats since most of the estimates are not very certain. So we did kind of some sensitivity analysis to look at that, and then and so once we had the oocyst loading, or the oocyst kind of layer across the landscape based on how many cats were where, then we put that through a hydrological model, and the one we use is called the Invest Nutrient Delivery Model, which basically treats oocysts as like a dissolved nutrient because the particles are so small that they're essentially acting like they're dissolved in water, and then that can be discharged with runoff. And the model takes into account elevation and slope, stream flow, annual rainfall, and land cover, so like how erodible different landscapes are and how much is likely to be carried off of those different landscapes. And so these are some of the maps that we came up with. And so as you look at these, the color scale is consistent across all of these maps. So like where you're seeing here for the feral and the stray cats are a lot more green to light yellow, whereas the colony cats and then the companion cats are getting more into the oranges and reds. That is a difference in the total expected oocyst export from these different cat types. And part of that is, so say with the feral cats, you've got cats that are, mostly up in sort of the Malka areas, the conservation areas that are further from the coast. So there's farther for those oocysts to go. So there might be a little less probability that the oocysts are making it all the way to the coast. But also, cats that are not getting supplemented with human feeding and just relying on catching their own prey are going to live at a much lower density. So we're also seeing, just fewer cats and farther from the coast with the feral cat category. So that has, kind of a lower expected oocyst export coming from those cats. Whereas like with the colony cats and the companion cats, those are living more in the human dominated areas, which, just given the topography of Oahu, tend to be closer to the, closer to the coast and on like that final slope where runoff is pretty easy. And the storm drain systems make easy runoff. So we're able to get more runoff. And also given the human feeding of those cats, we also have much denser cat populations showing up in the model as well. And then you can also see some just differences in different watersheds having, like especially these in central Oahu, these watersheds between the Waianaes and the Ko'olaus have really large catchments. So the potential oocyst numbers for export get pretty high in those watersheds, in part just because they're huge watersheds with a really big basin. So some of the conclusions that we could see from this project are that these human-associated cats really are sort of making the largest contribution to oocyst loading on the Oahu landscape, and particularly, with their habitat closest to the coast and the really high densities that they can reach. And then we also saw, we could see some of those large watersheds really kind of showing up as hot watersheds with a lot of potential runoff. But it is worth, reminding ourselves that like those big central watersheds that are, kind of running off mainly into like the Pearl Harbor area. Pearl Harbor is not necessarily the biggest monk seal use zone. And we are seeing Toxoplasmosis cases even up on the windward side where we didn't see the watersheds being as hot. So it's good to remember that with the relatively very small infectious dose that's required, like very few oocysts are needed to make a monk seal very sick. And then especially with that relationship that we saw with major runoff events, a good flush of oocysts from the land to the sea could still make monk seals sick, even if it's not in the, in the hottest watersheds that we found. And so some of the next steps with this, we want to take this model and really develop it into a tool that we can use to kind of run different scenarios through and really examine what sort of impacts on oocyst export we could have with different actions. But then there's also a number of other research avenues that are really important. We want to examine the genotypes of the parasite that monk seals get so we can understand, kind of maybe what cat populations or where on the landscape the disease is most likely to come from. And then also we just really want to focus on health assessments, especially for those adult females that are hardest hit by the disease. And we'll pair that with instrumentation so we can get better ideas of where the monk seals are going and what parts of the nearshore environment they're using the most. And then of course, also all of all of this research contributes a lot of data, contributes a lot of understanding, but it doesn't really mean much until we engage with managers and stakeholders and other partners so that we can really think about how to put all this knowledge into action to mitigate this threat for monk seals. And so Angela is going to come back on and talk to us some more about some of those activities.
Thank you, Stacie.
Oh yeah, and I'll stop sharing.
That's all right. Let me... Okay. Should be back up there. Hoping for no more technological issues here. All right. So how do we tie this into management? This is, again, a bit of a repeat slide from Tuesday, and I'll just run through it really quickly. Oh, sorry, I've got my cat joining me now. But some of the challenges that we've mentioned, and a couple that we may not have touched on previously in these presentations, there are a large number of cats. We don't necessarily know exactly how many there are or where they all are. Estimates for Oahu have ranged in the past from 50,000 to 300,000. It's a huge difference. We didn't really get into the law and policy side of things, but there are very few laws pertaining to cats. Most of them are county level, so there's not a lot of statewide consistency, and there isn't a lot of enforcement. People aren't checking every cat to make sure it's microchipped. There are steep fines for abandoning a cat, but you rarely catch someone in the act of doing so. Those characteristics of our environment, some of which Stacie talked about, are topography, heavy rainfall. Those are really conducive to toxo flowing through different habitats and ecosystems. Warm weather is also really favorable to cats. We have no large predators, so their populations grow quickly. As a lot of folks on the line might know, there is disagreement over different management strategies for cats that are on the landscape, things like trap, neuter, return, or humane dispatch. And so these disagreements can cause stalemates and prevent progress from going forward. Cats are not necessarily viewed in the same way that a lot of other domestic animals are, like dogs or even farm animals kept on leashes, kept in pens, kept in barns, kept in houses. And for some people, there's even a perception that cats should have unrestricted access to the outdoors. We talked about the challenges with seals. It's hard to detect it early. We don't have a lot of options when it comes to curing the disease or preventing it. There are no vaccines for monk seals or for people or for cats, although there is work being done. And lastly, for us at NOAA, we're an ocean resources agency. So we're facing this terrestrial threat. We have no jurisdiction over cats or the terrestrial habitats that they live in. So partnerships here are really, really important for us. So what do we do, in the face of all these challenges when we're looking at management solutions? So if we're looking to functionally eliminate T. gondii oocysts in the environment, that means not necessarily absolute zero, but that the rate and frequency of oocyst introductions are so small that they're essentially non-consequential. This is our ultimate goal. Then there's sort of 2 points of intersection here. Reducing the number of oocysts coming into the environment, limiting the transport of oocysts that are already in the environment. So if we look at this side, limiting transports of oocysts that's already in the environment, this has a lot less impact. So we don't spend a lot of time on this. There is no treatment for Hawaii's stormwater currently, but like Stacie mentioned, these oocysts are so tiny and so hardy. They will go through most physical treatments like filters. They're resistant to a lot of chemical treatments. They can survive up to 2 years or more, depending on the right conditions, even in highly acidic environments. So for those reasons and others, that's not necessarily a pathway that we're focused on. That's not to say that stormwater treatment is bad, just that in this case, it's not a priority. Restoration and creation of wetlands as well, creating sort of natural filters. Also a really wonderful thing for many purposes, but for this one, what you're really doing is sort of just passing the buck onto a different habitat or ecosystem. It could prevent oocysts from entering the marine environment, which is great for monk seals, but what about the water birds and other species that reside in those wetlands? So again, not a bad thing, just not necessarily the best solution to this problem. So we'll focus our efforts on this side, looking at oocysts coming into the environment. So in order to do that, we have to address cats that are already on the landscape and prevent new cats from coming onto the landscape. Because no matter what management method you're using, if you cannot prevent new cats from coming onto the landscape, then it's an ongoing battle. That in particular, this prevention aspect, if you were joined us on Tuesday, we talked about turning off the tap or the flow of new cats onto the landscape. This is a good point of collaboration. If we're talking about, this is less divisive because, for example, conservationists, cat welfare professionals share a common agreement that there are too many cats on the landscape, that an indoor lifestyle is better for cats, and they don't want to see abandonment. Don't want to see, dumping, support spay neuter. So this is a good point to focus on for collaborations, and is somewhere that we've been focusing a lot of attention. Definitely not ignoring the cats that are already on the landscape, and there are a lot of organizations that are doing really great work there that NOAA partners with and supports. But also, again, given our role as an ocean resources agency, not much that we can do directly on the ground. So again, we've been doing a lot of focusing lately on this prevention aspect. There are 4 sort of main factors or routes to reducing the amount of oocysts coming into the environment, looking at both of those subcategories there. Regulation and policies, those are laws and policies like microchipping, spay neuter, licensing, things like that. Enforcement is another, as I mentioned, education and outreach, informing the public, making sure accurate information is out there and available, educating legislators, making sure that there is an environment in which people are aware this is an issue and are supportive of implementing management and control measures, which is the final category. And of course, supporting all this with collaboration across different sectors, with the community, between agencies, and research. We know a lot. We know enough to act and to do things, but learning more always helps us act smarter and act more effectively. And so ongoing research is important in order to be able to help us do that. We've made a lot of progress since 2015. We have gotten our social scientists at the Pacific Islands Fisheries Science Center engaged. Kirsten Leon spoke on Tuesday for those of you who were able to join us. And we've learned a lot there about tackling this problem from a collaborative, understanding, core values perspective. And I think that's helped a lot in the work that we've been doing. We have an interagency working group, the Toxoplasmosis and At-Large Cat Technical Working Group that was founded about 6 years ago. It was initiated by NOAA and DLNR, and we continue to meet quarterly. We share resources and knowledge. We have conducted our own outreach. And we're currently working, a subset of that TACT working group is currently partnering with the Hawaiian Humane Society on those things that we can do that address controlling the source of cats. Things like counting cats and understanding where they are and what types of cats they are, because we both acknowledge that you're not going to manage a feral cat way up Mauka near a seabird colony the same way that you are going to manage a stray cat wandering around downtown. We're working on messaging campaigns about Pono pet practices, Pono pet parenting, primarily keeping cats indoors, which is good for wildlife and for cats, gives them a longer, healthier life. And hopefully by doing this, we're building trust to garner broad support for a multi-pronged statewide strategy, just meaning that there is no one silver bullet solution. We cannot implement the same thing on all islands in all communities and expect it to work. There's going to have to be a lot of different things going on that complement each other in order to achieve success here. And with that, before we go on to questions, I feel like I really just want to acknowledge the really unfortunate passing of Fern Duvall. I don't know everyone who's on the line today, but I know a lot of you knew and loved Fern. And sorry if you didn't know, this is a topic that Fern was just really passionate about. He worked really hard on for decades. The last time I saw Fern in person was at a toxoplasmosis workshop about a month or so before COVID hit. He was a wonderful mentor for me when I was at DLNR, and he's just been an amazing colleague and friend, I know to a lot of us. And we wouldn't be where we are today as far as making progress on this issue without Fern. So apologies for those of you who weren't already informed, but I just felt like it was important to take a moment to acknowledge him. Thank you.
Thanks, Angela. Sorry that I had not heard that. That caught me off guard a little bit. Thank you for that acknowledgement. That's a great loss to the Hawaiian conservation community. So, going into the Q&A, there was a couple of questions. Stacie, maybe you could reference, there was one in the chat. At some point you had asked about, or you had mentioned a paper that you had referenced several other papers about, the amount of poop that cats make and how many cats are infected with toxoplasmosis. I'm kind of thinking maybe you have a paper that has all those references in one that you could share, or you have some other way to point folks towards those resources.
Let's see, I myself don't have a published paper, but we do have like a research plan government report that compiles a lot of that literature. So I could share that, or if folks are interested, I could share just like a couple of the like literature summary tables from that. I don't think I can handily find it just right now to drop in the chat, but I can definitely follow up and provide those.
That would be great. Yeah, and I think if you logged in using your email, I will be able to follow up with the Zoom and be able to email folks. So if folks logged in using their emails, I can follow up with any additional resources. Molly, do you have any questions over on Facebook?
There are no Facebook questions.
Okay. Jade, what do you have in Q&A?
There's a couple. Is there a blood test or something for cats you can tell if they're infected? They already have the toxoplasmosis.
The answer to that is sort of, and I think the numbers are something like by the time cats are 2 years old or reach adulthood, I mean, the infection rate is nearly 100%. But here's the trick. Cats get infected, but they're not shedding all of the time. They're mostly shedding shortly after they're infected. They can, if they get infected with a new strain later on in life, they can shed again. So the blood tests, which typically test like blood serum, like when we get our serology tests for different illnesses, it mostly tests to see if your body has made antibodies for that pathogen. So that basically says at some point in time, your body encountered this pathogen and made antibodies. But with cats, it's like, so basically just all of them look positive on serology because eventually in life, almost all cats encounter toxoplasmosis, but they're not necessarily shedding oocysts all of the time. So they can do fecal tests for that, actually test the cat's feces and do a float to see if the little oocysts are floating in there. But that can be a lot harder to get a sample if you just have a cat in hand for a moment.
Okay, thank you. And I got 2 more questions. So like, is there research, or if there's, how is it going for getting a vaccine for cats against toxoplasmosis?
So there is ongoing research, especially in Europe, going on into this right now. And nothing so far. I think on Tuesday I mentioned that New Zealand has a vaccine for sheep, but it's a live vaccine, and we cannot use that in the US, and it doesn't work on any other species. So as of right now, it's not really an option, but there is work being done on it. One thing to keep in mind though about a vaccine is that vaccinations might prevent the risks from toxo, but they wouldn't prevent risks of predation, which we didn't really talk about, but cats on the landscape are also predators. And the logistics of administering a vaccine, I mean, it's hard enough for us to get an idea of how many cats there are, let alone catch them all and administer a vaccine. So not to discourage the vaccine or a vaccine, it would still be wonderful and useful and a great tool, but it wouldn't be it still wouldn't be a silver bullet solution.
How do the oocysts actually affect the Hawaiian monk seal? Like, are they eating it when they're feeding near shore? And also, are the Mediterranean monk seals affected?
Sure. So I think what you're asking is how they get into the monk seal in the first place. And I think that's one thing we didn't cover, which is so cold-blooded creatures can intake the oocysts, like filter feeders or fish, but they won't get infected. So they act kind of like little banks, like little repositories of oocysts. So if a monk seal eats an infected prey, then they could get it that way. They could also potentially just encounter it directly in the water. And as far as Mediterranean monk seals, Stacie, there's been one documented case.
I believe I've heard of it happening in Mediterranean monk seals,
Yeah.
but not nearly with the frequency or intensity that we've seen it with Hawaiian monk seals. And whether that's because in the Mediterranean they might have evolved more closely with cats and been exposed through longer evolutionary time, whether it's because in the Mediterranean they manage cats differently and don't have as much toxoplasmosis going into the water, or because Mediterranean monk seals are, even fewer than Hawaiian monk seals and spread out amongst, thousands of islands within the Greek Isles and the Mediterranean Sea, and potentially they're not detecting as much. So there could be a number of factors there, but it's certainly possible for them to get it too.
We have another question that just came up on Facebook that's somewhat similar. Does toxoplasmosis, have there been increases in other crustacean species as well, like whales and dolphins?
Yeah, this is a very big, just to give one example, short answer, yes. And one example is the Hectors and Maui dolphins in New Zealand. That's another example of threatened and endangered species. They're two different subspecies. And another example where females are being disproportionately affected, and where that's become of increasing concern. So New Zealand has been very focused on that lately as well.
Okay, does that sum up the questions from both sides? Actually, there's one interesting I just saw in the Q&A about could maintaining feral cat colonies doing TNR be seen as a violation of the Endangered Species Act now that we have information about the impacts of this on an endangered species?
So there's only one, there's only one cat-related case that I know of for sure off the top of my head, which was American Bird Conservancy in the state of New York. And that was where a cat colony was, and Stacie, jump in if you remember. I can't remember if it was actively being maintained by the state or if it just was not being, or if maintenance of the colony wasn't being discouraged by the state. But it was on state land. It was near to a plover colony, an endangered bird, and the cats were eating the birds. So there was a lawsuit, and it was settled out of court, but the colony was removed. The closest example, and that was an endangered, under the basis was that it was violation of the Endangered Species Act. The closest case I can think of in Hawaii was with Palila and the goats on the ungulates on the Big Island that were causing damage, and that was also an Endangered Species Act case. So there's precedent, but not necessarily a lot of it, but there is precedent for it.
Interesting. That will be interesting. Oh, one more. Do oocysts shed any DNA so that eDNA surveys could be informative? It's kind of a really cool technology. Stacie, you're nodding.
It is a really cool technology, and that's definitely been an idea that has been thrown around. We actually worked with a student a couple years ago at UH that was doing, doing some trials, getting eDNA from different harbors. I think they had a little bit of success. I think one of the challenges is that a meaningful dose of oocysts can be as little as a single oocyst. And then of course you're putting these microscopic things into the ocean. So it's like, it's so much harder than a needle in a haystack. It's literally like a microbe in an ocean. So I think it's certainly possible. They do contain DNA. I think labs have to use some special DNA extraction procedures to kind of get the DNA outside of that really hardy oocyst sort of shell. But that's certainly possible. But I think what has been tricky so far is just like getting the amount of filtering or whatnot to get a good sample where you're like, how much water do you have to filter to potentially have captured some oocysts? So I think the general answer is it is a really cool technology. It is totally possible. It's not easy, but I think it's possible.
I found that really fascinating, the idea that just one oocyst. I mean, I work a lot with rat lungworm outreach, and it's the load of parasite is actually really important to the, or they think right now that the load of parasite is actually important to the severity of disease, that the more parasites you get, and this sounds like you don't need a load of parasites, you need one.
Well, and we should say we don't necessarily know what loads the monk seals are encountering when they get sick, but so the, it can take as little as one oocyst is kind of more from clinical veterinary research, more generally. But given that monk seals are wild animals and we're not, studying them in controlled exposure experiments or anything like that, we can't be sure. But we know from the literature that it doesn't have to be a lot, but we also don't know, how big of an influx they need. But, you can imagine more oocysts equals more probability of encountering whatever that necessary number is.
Molly, was there another question? No? Oh, okay, maybe. Okay. All right. Well, so much great information that you presented here. Really appreciate it, and appreciate everyone for tuning in. Again, I will reach out to the folks who had their emails to sign up for this, and Stacey's going to share some follow-up information so I can get that out to you folks. But really appreciate you being here for this and learning more about these threats to our monk seals, and thanks to our speakers for giving us such great information and transparency into this work, so we can really see just how it's just the steps that you have to take, in science to really gather all of this information and build that case, like what is going on out there, and you guys are doing the hard work. So thank you so much. Much appreciated. Aloha, folks.
Thank you, Franny.
Thanks, Franny. Thanks everyone for joining.
Turning off the recording.